Well, good afternoon, everyone. I'd like to welcome you to today's webinar, which is being co-sponsored by the U.S. Forest Service and the Association of Fish and Wildlife Agencies. My name is Jonathan Maudsley. I am the Science Advisor at the Association of Fish and Wildlife Agencies. And this webinar forms one in a regular series of webinars that we host in collaboration with our partners at the U.S. Forest Service and the Research and Development Program. Today's webinar will be looking at genetic technologies and is entitled What Natural Resource Managers Should Know About Genetic Technology Terminology and Techniques to Understand the Considerations of Value versus Risk. A great presentation today from Dr. Daniel Lindner, who is Research Plant Pathologist, U.S. Forest Service R&D at the Northern Research Station. Before we get going with the webinar, I did want to go over some brief housekeeping. We will be muting all of the lines except for Dr. Lindner's line. But we ask, if you do have your phone, we ask that you mute that as well if you have that capability. We will usually bring the mutes, we'll turn off the mute at the end of the webinar so that we can have a question and answer and discussion section as time permits. And at that point, it's sometimes distracting if people have their phones on and are typing. But we also invite you to submit questions during the course of the webinar. We have a chat box in the lower left-hand corner of your screen. I've already put one note out there. There will be other ones coming just to remind you to, if you have questions for the speaker, to please feel free to type them in that box and send them in. I will have a list of those, and then when we get to the end of the presentation, we'll start with those questions first, and then as time permits, we can move on to a more traditional question and answer where people ask questions over the phone. This webinar is being recorded, and it will be available on the Association's YouTube channel at some point after the webinar is complete. That channel can be found by going to YouTube, the website YouTube, and looking for Association of Fish and Wildlife Agencies. We have recordings of a number of other webinars that we've conducted with the U.S. Forest Service as well as other federal and state partners. So at this point, I would very much like to turn over the webinar to my good friend and colleague, Dr. Monica Thomasy at the U.S. Forest Service, to provide us with an introduction and background to today's talk. Monica.
Thank you, Jonathan. Thank you for this opportunity to connect biologists and managers with some more of the work we do in the Forest Service Wildlife and Fisheries Research Programs for the wildlife and fish management community. Through this Association of Fish and Wildlife webinar series, we are able to address many questions that state agencies and other biologists have on current research being conducted, results of completed research, and scientific topics of current interest. Today's topic, genetic technology, was identified as a result of the AFWA Science Committee's curiosity understanding new techniques that can be used to alter DNA and the implications to wildlife managers. In response, today we have Dr. Dan Lindner, a mycologist who studies ecology and genetics at the Forest Service's Center for Forest Mycology in Wisconsin. Today he will provide us with some basics of what we need to know in order to have useful discussions on the management issues and the scientific information needs. And he'll provide an illustration of how genetic technology could be used to address invasive species issues. Dan is a mycologist. His research examines how human actions can affect fungal communities and how the resulting changes can affect ecosystem function. For about 10 years, he's been working with many wildlife biologists due to the outbreak of Whitenose syndrome, which is caused by a soil-dwelling fungus. I appreciate Dan's willingness to speak on this topic of genetic technology, especially since he's produced foundational information on the genetics of the Whitenose syndrome fungus. Dan has a Bachelor of Science degree in botany and a PhD in plant pathology, both from the University of Wisconsin in Madison. Thank you, Dan.
Great. We're going to bring down the lines now at this point and bring up Dan.
The conference has been muted.
Thank you, Monica. Well, thank you, Monica, for that introduction, and thank you for having me here today. It's my pleasure to be talking on this subject. And I want to start out by saying that it is an incredibly complex and broad subject. I don't pretend to be an expert in all of the different aspects of what I'll be talking about today. And I really see this as sort of the beginning of a conversation about this, and that trying to get people to the point where they can ask questions and have informed conversations. But even myself, who works in this field specifically for some of the things, trying to remember and keep up with all of the details is incredibly complex because there's so many different techniques and the techniques change so quickly. For some of these things, even defining the concept of value and risk, these are things that we could spend a huge amount of time on. So I'm going to give an overview to do today, and I'm sorry if I don't focus in specifically on some topics as much as people would hope for, and if that's the case, feel free to follow up, and through email or other things we can have longer discussions about specifics. But let's see, I guess, can we advance to the next slide then? My take as I approach the idea of using these new genetic technologies for whatever purpose, but in specific for thinking of it in the context of wildlife management, is that you have these kind of usual trade-offs of looking at how well does something work versus how much risk are you taking on. And next slide, please. That should bring up what it is that we hope for. There in the lower right corner, you always hope for something that's going to work really well and have low risk. And of course, what you don't want is what we see that X in the upper left corner, something with a lot of risk that works quite poorly. And what we usually get, next slide please, is inevitably something kind of along this line, that we can find a lot of solutions that are maybe low risk, but then they don't work very well, and we can come up with other solutions that could potentially work incredibly well, but then are correlated with a lot of risk. So as I've thought about this, and I'm sure other people have identified this, these things to be the typical trade-off in this kind of work, and that maybe someday we'll get to that lower right corner, but for right now, a lot of the solutions and things that I'll talk about, you kind of have that trade-off. You can have something that works. You can have something that works very well and is often associated with higher risk, or something that has low risk but tends to maybe not be a perfect solution to the problem. So next slide, please. Obviously, as most people are well aware, risk is never going to be, you'll never be able to get it down to zero. And I know there are people who study risk and all of these things and the definition of risk and risk types and value. And so I'm just going to go over this quickly, but saying, just being aware that not doing anything also comes with risks, that it's not easy. Doing something can cause problems, but not doing something can also cause problems. And that as scientists, scientists tried to provide info so that managers can evaluate and decide what to use, but particularly in this field of genetic technology, there often is this disconnect because of there's so much terminology, and the techniques can be so confusing or full of technical details. And these technical details change on certainly a weekly basis, if not a daily basis, and sometimes it feels like by the minute. So, but that's, one shouldn't give up hope. I feel like if you can understand some of the terminology in general and some of the overview of the techniques, it's a gateway to being able to get into this conversation of what is risk versus value, and to be able to start asking questions. Next slide, please. So in terms of terminology, there's 30 or 40 things easily that one could list. I'm just going to come up with 4 sort of general things that I listed here today that are good background things that I want people to know, while also recognizing that some of these things, like the second one there, DNA versus RNA versus protein versus genes, I'm going to assume a little bit of background knowledge there. It's too much to cover in a webinar like today, but I'm going to try and keep a lot of it at a very broad level, at this sort of broad level of that DNA is instructions. And RNA these days, when I learned about RNA, there were only 3 types. There are so many types of RNA recognized currently. I can't keep up with them, and RNA does many, many different things. But we can often think of it as something that regulates and passes messages, and that the central dogma of biology in its simplest form is that you go from DNA to RNA and RNA to protein, with protein being sort of the building blocks of biological cells and organisms. To skip back to that first one, when we talk about genomes are just simply the sum total of all of the DNA that an organism has. And then to skip down to the third one, a term that often comes up is gene vector and plasmid. And a gene vector in the simplest form is you can just think of it as something that we use to move DNA around, a vehicle to deliver DNA typically to a foreign cell. And when we say plasmid, a plasmid is simply a small circular bit of DNA. These small circular bits of DNA tend to be easy to move around, but a vector could also be a virus. You can sometimes use viruses to shuttle DNA around, or it could be a full artificial chromosome, a larger chunk of DNA, not just a small simple circle of DNA, but a larger chunk. But no matter what it is, it's just some way to move. When we talk about vectoring something, there's a variety of different vectors, but it's just a matter of trying to move certain pieces of DNA from one place into other cells. Now, the most critical thing here, I think, or as I was thinking about what is it useful to know, I think it's important to remember this definition between somatic tissue versus germline tissue. And you'll sometimes hear different terms for this. Germline tissue is sometimes just simply referred to as reproductive tissue. And somatic tissue is body tissue in the simplest form. If you're thinking of animals or vertebrates, I work with fungi typically, and fungi it's a bit more complicated, so I won't go into that. I'm generally going to be thinking in terms of vertebrate models for the most part here, and that somatic tissue is your body tissue, anything that's not going to be a cell that has the potential to be passed on to the next generation. Whereas germline, if you're a germline cell, that would typically be something like a sperm or an egg or a cell that produces one of those things, and a change in that kind of cell has the potential to be inherited, to move on to the next generations. And I think when we talk about these different techniques, it's important to remember, are they affecting somatic tissue? Are they affecting germline? And to get at some of these questions. And next slide, please. I've got some sort of example questions that I'm just going to throw out here at the beginning, and then by the end you can see all of the types of questions that you may have by the time we talk about more of these techniques. But questions that I can think of that when I approach a technique, some of the first things that I ask are, does the technique affect the germline? Is it something that's going to be heritable? Or does it just affect somatic tissue? Are you doing something that's just going to affect that particular individual and you're not looking, or that you're going to apply it to populations of individuals, but you don't actually see it as something that's going to be passed on to future generations? Other questions are things like, once it's deployed, will the effect be self-sustaining? Again, that often comes back to this question of, is it in the germline or is it in somatic tissue? And another question that comes up is there a kill switch, or is there some way to reverse the change? And inevitably, I would say for most techniques that we come up with, if it's in the germline, rarely is there a kill switch or some useful kind of kill switch, although that is something that people have talked about trying to build into many of these techniques, so that even if you have something that gets into the germline, is there some possible way to potentially reverse it? Other questions then evolve more around sort of non-target or off-target effects, such as these changes that you make, can you really ensure that they're going to be limited to the target species? What is the chance they could get into other species? So these are just an example of some of the questions that start to come up. So next slide, please. So now we'll go through an overview of some of the different techniques that when I was thinking about this and trying to put this together, thinking what are the techniques that come up most frequently. And I'm going to lead off with gene and genome editing. And again, I've got that caveat at the top, these things change daily. Just to remember that these techniques are constantly being tweaked. although many of the overall categories of techniques are more or less the same. So if you can understand the sort of category of technique that you're working with, that may still hold true a year from now or 5 years from now, even if they've tweaked the technique a bit. That it may still be a gene or a genome editing technique, even if they're now using a different enzyme or some other way to do it. And I will say that gene or genome editing is really a subset of this broader group of genetic engineering techniques. Anything in general doing genetic engineering, genome and genome editing has come to the forefront recently because of CRISPR-Cas9, which I'll mention as an example. But the definition of genome and genome editing is really comes down to just this ability to selectively and specifically change or edit the genetic code of an organism in a really targeted way. We've been able to add things to genomes or do genetic engineering for quite a while, but it's been rare or it's been extremely cumbersome to be able to do it in a very specific, directed way with that. So in terms of examples, there are things like CRISPR-Cas9 that people may have heard of. There's also techniques like TALEN, zinc finger, the DFNs, the zinc finger nucleases. There's actually 4 main categories. I've left one off there, but there are different ways to do it. The reason that CRISPR-Cas9 has gained so much attention is because it's so easy, cheap, and fast. It's not that we haven't been able to do these things before, but in order to do them previously often involved an entire PhD just to do a small change. It was something that involved a vast amount of resources, and it involved a commitment of time. So it was the kind of thing that governments or large groups would do. CRISPR-Cas9 really has the potential to be something that could be done cheaply, efficiently, easily, quickly, in a lab that could be set up quite quickly. And it's not quite stretching it to say it's the kind of thing that with $8,000 to $10,000 you could set up in someone's basement and be coming up with a system to be at least playing with those things. Whereas previously you were talking about if you were going to do it, you would have to have a lab where you're investing hundreds of thousands of dollars in infrastructure just to even be able to begin to do some of these techniques. So that's why CRISPR-Cas9 has really grabbed people's attention and the media's attention, because it allows scientists to, it will exponentially increase the speed at which things are developing in this particular area. So I think that covers it from there. So next slide. So the uses of gene and genome editing, I tried to come up with an example that people may have heard of. The most obvious example that I could think of was this idea of editing black-footed ferrets to be resistant to sylvatic plague, the same sort of black plague caused by Yersinia pestis. This is not my area of expertise. I do not know that much about it, but my understanding is that domestic ferrets or other types of ferrets are immune or do not get this disease, and the idea is to do comparative genomics so that you can look at the genome of the ones that are resistant, compare that to the black-footed ferret's genome, figure out what that difference is, and see if you can introduce that difference through genome editing into the black-footed ferrets. The one group that I believe is working on this, that I know is working on this, is Revive and Restore. There may be other groups that are also working on this. You would have to move it. It's complicated, obviously. So that's the simple view. In terms of risks vary greatly and are strongly based on whether edits are in somatic tissue or germline. If you're just going to do this in somatic tissue, you'd be making one individual perhaps resistant, and the goal, as I've read it, is to move it into the germline so that you could create a line of ferrets that was resistant. But of course, the genetic pool of black-footed ferrets is already quite small. So the problem is, or one of these sort of complicating factors is, you can't necessarily just move it into one ferret or one genetic background, in that then that one genetic background needs to be sort of mated with the rest of the population in order to get this phenotype expressed throughout the population. And so you would actually need to know this in all likelihood, not just to one ferret, but to as many different genotypes of ferrets as you possibly could, create cloned ferrets and have them interbreed or mix with the population. That's the idea, at least. And I would say, again, in terms of these risks, for that particular example, you're looking at something that you are affecting germline cells. You're looking at something that would be propagating throughout the population. So I would say that that is something where you have the potential to have very highly, be very highly effective, but you also are taking on a great deal of risk. So gene and genome editing. So with an example and some of the risks, next slide, please. Now, the next thing so next to CRISPR, I would say the other thing that I hear the most about in the news are probably gene drives. Gene drives are something that you can set up with CRISPR, and it's simple. Trying to understand how one creates a gene drive is quite complex. And I'm going to leave the definition that it's a genetic modification designed to spread rapidly through a population. And I threw sexual in there because we'll get back to that. It has to be a sexually reproducing population. And it's this trick you use to get around Mendelian inheritance laws of that, using sort of selfish DNA ideas of how you can get something so that instead of 50% of the offspring having it, that you increase your odds, or pretty much bias it totally toward whatever it is, so that this gene or trait will be driven through the population, and that you can make it so that there's, in theory, 100%, or in more realistic, maybe 90% of the offspring will have that trait instead of just 50% of the offspring. So in terms of gene drives, examples where this has been done successfully and has been shown, it has been done in some insect populations. Fruit flies were one of the first. It's also been done in fungi. It's been done in yeast populations. It's then also been moved into mosquitoes. My understanding is, I believe it was around February, that they announced that they had the first mice that had been engineered in this way to potentially have this gene drive. Maybe other people have kept up with this more. I'm not certain if that's been moved to the point where they've been able to show that it functions in those mice, that it's actually passed on to their offspring in the way that they would expect if it's truly acting as a gene drive. But it's definitely within this since February, gotten to the point that scientists have created mice that potentially have this molecular machinery in them, this motherless technology, which it's again created through this CRISPR system, this gene editing system, and it's motherless is a particular gene drive that skews sex ratios, so that you pretty much just have males. And as the population heads towards more male and male dominated, obviously that's going to cause population crash or extinction. Next slide, please. So the use of gene drives is that the idea is it could potentially drive a species to extinction, and if nothing else, at least make populations crash. Limitations, as I mentioned, sexual recombination or sexual reproduction is rather vital to make gene drives happen. So populations must sexually reproduce in plants. This doesn't always, if there's a lot of inbreeding or self-crossing, that can be problematic. And it won't necessarily work in things like viruses or bacteria, which are truly asexual. And for fungi, it's very complicated. There are some fungi that do reproduce sexually and outcross quite frequently. There are others that stay asexual for long periods of time, so it wouldn't necessarily work there. In addition to requiring sexual reproduction, gene drives really require fast generation time. In general, the theoretical calculations show that it's about 12 generations until you reach a significant, that the proportion of the gene you're trying to drive through the population until it reaches a significant fraction of the population. So 12 generations for humans or elephants is a big deal. 12 generations for fruit flies or mice isn't necessarily as much of a big deal. So if you've got fast generation times, this can happen. And you can also augment this by the initial release size. So this initial release size, if instead of just putting one or a small number of individuals out there with a gene drive in them, if you seed into the population every 1 in 1,000 individuals has the gene drive, or 1 in every 100 individuals has the gene drive, then it will spread through the population much faster. But this is also based on gene conversion rates and that kind of thing. But the bottom line is that to really make it work, populations need to reproduce quickly. Next slide, please. Gene drives continued, risks and advantages to work. Again, they must be in the germline. Thus, it's inherently that you're taking on some inherent risk there because it can be self-propagating. The whole idea of a gene drive in order to have it work is that it needs to be self-propagating. And other risks involve the fact that species interbreed or sometimes hybridize with close relatives. There's also this question of accidental release. Once you create a species that has this gene drive in it, what happens if you, so like zebra mussels, maybe you want to do it in a certain area, maybe other areas you wouldn't. If it's released into its native range by accident, you may wind up wiping out the population itself. So how with mice, when you talk about releasing them on islands, the beauty of mice is they're incredibly good at getting around. So you believe that your genetically engineered mice are on that island only, but we've all seen the pictures and cartoons of rats and mice climbing ropes and getting onto ships and all the different ways that they can get around. So what if it accidentally got back to its native range? What if unwanted mutations also occur and then tag along with the gene drive? You've now created this drive that makes things move through a population quite quickly. What if a mutation occurs and something starts to tag along that you really never expected and you really don't want? You've got a gene drive, and the gene drive for the motherless technology mutates and doesn't work, but it accidentally got attached with something that makes the mice super susceptible to fleas. So now you've driven a gene through the population, just as an example off of the top of my head, that makes mice super susceptible for fleas, so now they're able to transmit fleas at higher rates or something like that. So that's just a complete theoretical thing, but this idea that you could have unwanted mutations that might tag along with the drive. Advantages, of course, are based on what is one of its major risk factors, the fact that it's self-propagating. The problem with human treatments that involve inputs to try and go after a pest species, is that we often can't reach all of those. We can't access all of those areas that these creatures get to. So a self-propagating solution is the only kind of solution that may work in some of these situations, and that is what is the big draw, that they could work, say, on an island where, if you know, there are no non-native pest populations around, but there's crags and peaks, high altitude, a lot of small caves and crevices and cracks that these creatures go into. It gives you the ability to have it move through the population, assuming that the population is well mixed. If the population doesn't mix, that's another problem. But at least in theory, that's one advantage of a gene drive. Next slide, please. Risks continued. I'll go on here with the risks to gene drives just because I think this is so important, and I'll even include some stuff with really small font. I apologize for that, but I think this is important enough that I'm actually going to not only put it in small font, but read it, because I think it's definitely something worth going back to. And it's just from the summary of the National Academy's report that was titled Gene Drives on the Horizon. It's available as a PDF for free. I believe it's for free, that people can just download it. A great document, a relatively long document, so it will take some time to look through, but I think even just reading the summary is very useful. And this is some text lifted directly from the summary, saying proof of concept experiments and a few laboratory studies is not sufficient in and of itself to support a decision to release gene drive modified organisms into the environment. Laboratory and field research is needed to refine CRISPR-Cas9 based gene drives and other gene drive mechanisms and to understand how gene drives might work. This is what I think is particularly important under different environmental conditions and in a wide variety of organisms. The considerable gaps in knowledge about potential off-target effects, and when they say off-target, they're referring to within organisms. So like, again, the mutations tagging along with a gene drive, as well as non-target effects affecting other species in the environment. These effects necessitate a collaborative, multidisciplinary approach to research, ecological risk assessment, development of public policy, and decision-making for each proposed application of a gene drive technology. I would say that with a lot of this, we're really in the developmental stages. We're in the proof of concept stages, and that we've got a long way to go before we could sort of realize this idea of having more of this field-based information in place. Next slide, please. So then, a general term, sometimes synthetic biology is applied very broadly to almost all of the techniques that I'm referring to, but it's also more recently has kind of taken on this flavor when people talk about synthetic biology to mean designing and constructing new biological entities, synthesizing large blocks of DNA, perhaps from extinct creatures, from extinct DNA, where we know their genetic code, but we no longer have any living representatives, and using genetic manipulation, such as synthesizing the chromosomes, putting them into different eggs, finding surrogate parents, something that will work close enough, and trying to revive and restore things like the heath hen, passenger pigeon, woolly mammoth. These are all projects that are underway. To me, I see these as this. I sort of put up. I still sort of see this as a curiosity, and maybe that's wrong of me. Maybe these are closer, but in my opinion, I view these as slightly further out. The technological hurdles that need to be overcome to make these really successful, I think, are still quite significant. Whereas, say, gene drives, gene editing, some of these other techniques, we're definitely there. We're at proof of concept, and a lot of that stuff could be launched in the next couple of years. For some of these examples, I think it's perhaps a little bit further off. Heath hen, again, something as not a wildlife biologist, as a mycologist, something I know very little about, but the idea, my understanding is it's an extinct bird that had been in the southeastern United States. Could you bring it back? Could you bring back populations that were viable to provide the ecosystem services that these creatures used to provide? Passenger pigeon, hard to even imagine given the continental scale cost, which they, could you move it from something more than a curiosity of, I could almost see truly being able to, given that they did not go extinct that long ago, there's very good DNA available. I could see it getting to the point where you're able to create a passenger pigeon, perhaps enough to have a breeding colony. How you move that then to enough to get a self-sustaining population for a creature that had population density issues, and it thrived in these absolutely enormous numbers. I don't know. I have trouble imagining how that would move past sort of, again, a curiosity stage. But again, maybe I'm just limited in my my ability to see those kind of things. And then things like woolly mammoths, which people are working toward, and yet these are creatures that have been extinct for far longer. And I think there's more debate there in terms of why do that? Does it even make sense? Is there an ecological role for these creatures which were perhaps designed in some ways for the ice age or different environmental conditions? What would be the point? Would it simply be to put them in zoos? Or would it actually bring back a species that would perform an ecological function that made sense? So the uses, reviving extinct species, or potentially even just reviving species that are extinct or near extinct, and the risks I can't even begin to quite imagine, but I'll just go with the assumption that everybody has seen Jurassic Park or is familiar with it, and you can use that for fodder for your imagination of all of the different things that could potentially happen with bringing back extinct species. Not that I necessarily think the heath hen would become a terrible predator of humans if it were brought back. But all sorts of ethical and interesting issues there that could be addressed. Next slide, please. then I'll talk about gene silencing, and I'll go through an example of this because gene silencing is one of the techniques in particular that my lab has worked on. We've tried to use this in relation to Whitenose syndrome of bats, so I'll use that as the example. And the definition of gene silencing is you're trying to reduce expression of a particular gene. In the next slide, I think I use the term knockdown rather than knockout. We sometimes refer to gene silencing as knockout. You try and knock that gene out, so it's no longer functioning. But it's not quite, there's different flavors of it. You can sometimes just knock it down, not necessarily get rid of it or stop its function or expression, but just make it express less, or perhaps even manipulate it in a way so that it's overexpressed. But it's a way to reduce or manipulate expression of a particular gene. And as I say, example, Whitenose syndrome, next slide, please, that the use is knock out or knock down a gene that causes disease. This can be done using CRISPR technology. It can also be done with another type of technology referred to as RNAi or RNA interference. That's actually what we're using. Risks depend strongly on whether you're silencing genes transiently in somatic tissue, which can be done to creatures. And it's especially, that's how it would be typically done in therapeutic uses for humans, where you might be trying to control a genetic disorder in a human, and you may only care about it for that particular individual. And you could create their somatic tissue and perhaps reverse a disease within an individual. But there are also ways to do this where you insert it into the germline, and it then gets passed on to the progeny. In addition to directly inserting this into the DNA, there are ways to put this into a virus, where the virus then may move along with the organism. And if that virus continues to be associated with the organism, it's the equivalent of moving it into the germline. So next slide, please. So this is my example of gene silencing in Whitenose syndrome of bats. For people who aren't familiar with Whitenose syndrome, it's caused by a fungus, Pseudogymnoascus destructans, that I'll try, long name, so I'll just try and refer to it as PD. And it causes terrible disease in hibernating bats in eastern North America, threatening multiple species, not just one species, but multiple species, little brown bat, northern long-eared, pipistrelle, or tricoloreds, with extinction or at least regional extirpation. The goal is to silence genes of the fungus and to target genes that are referred to as virulence factors, things that this fungus needs to cause disease, and we're trying RNA interference. Next slide. So the first thing you have to do, I'll just run through this quickly, is that you have to identify the genes that you want to work with. And RNA interference, and again, it's too complicated to get into the details, but you introduce small pieces of RNA starting on the left side of this diagram. It can be done in a bunch of different ways, and in the end, really the output is critical genes are turned off. I'm going to leave it at that and sort of skip past this to the next slide now. And say that we're using our vector of how we're moving it. We use a vector which is a plasmid, and it's actually put within a bacterium called agrobacterium, sometimes just referred to as agro. This is a beautiful bacterium that's good at moving DNA around, originally isolated from plants, but also works with fungi. We use it to move gene silencing vectors into the fungus. We're also trying CRISPR-type approaches. So far, we've come up with 4 constructs that we've been moving into the fungus. We've had very low success rates in terms of transformation, but one of the genes that we're targeting is a spore production gene, so that we're trying to knock down how many spores this fungus produces, since the spores are the infective propagule in this disease. Also trying to knock down secondary metabolites at a broad level just to see what this could potentially do to the fungus. And then knock down melanin production. Melanin is a brown pigment. And has been hypothesized to be a virulence factor, something that fungi need to cause disease. It's been hypothesized that melanin is very important. So if you knock down or knock out melanin production, could you make a less pathogenic version of this fungus? And then at the very end there, with no arrow going to it, showing that we're also making a cassette or a vector that has Cas9 in it. So trying to move in the machinery that you could use to make CRISPR-like technology work. So once Cas9 is in the system, then find a way to introduce guide RNAs into those cells to create site-directed mutations. So that once Cas9 is integrated into the fungus, that would give us the ability to start using that sort of CRISPR framework of making really site-directed edits into the fungus. So trying kind of a variety of approaches here for how we could potentially go about silencing some of these genes in this fungus. Next slide, please. How is it applied? And the real question I would ask if I were you is, does it affect germline cells? And I would say it comes down to it can be applied in different ways. It can be done at the local scale, and you could just literally spray these types of RNAs, these small RNAs, on to bats or the environment, and it might have a transient effect that wouldn't affect the germline, but might be able to help save the bats because the fungus, it would knock down the fungus's ability to grow or do things. Alternatively, these gene silencing things could be integrated into both the genome or into viruses that pass throughout the fungus, and this could be important at continental scale control with the potential for being able to control the disease with limited human input, but again with the potential for long-term effects, and could be passed on through generations of the fungus. Next slide, please. Advantages, risks, and uncertainties. I want to be finishing up here shortly, so I'll just go through some of this, but say in this example, when I start to think about advantages, risks, uncertainties, I would say that we have the potential to be very specific about the gene we choose to try and silence. So ideally, we could target a gene or a gene segment that's very specific to this fungus and no other fungus. And I would also say that these oligonucleotides, that is, these short RNAs that we come up with, they're cheap. They're also generally quite non-toxic, and the idea is that they break down quickly in the environment. But then the reverse argument, sort of devil's advocate argument for both of those two statements that I just made, is that even though I said it could be specific, specificity is often based on what we know in databases. And most fungi are undescribed. We estimate only 10% of fungal species are described. So you could wind up affecting other species you don't even know about that are closely related. So non-target effects are still an issue just because we know so little about what we know is based on what we know, what we have in databases. But we know that there's a lot of unknowns out there, that when you've only got 10% of fungal species identified, 90% of them out there, there's a lot of potential for non-target effects. And then also the question of, do these small interfering RNAs actually persist in the environment? We know under more ideal conditions they can be stable up to 2 years. We don't really understand how they persist in the environment. So if you decided you wanted to get rid of them, do they really break down quickly, or is it something that might stay in the environment for a long period of time? Then based on the gene chosen, could you have unintended health effects on bats? These are all things that would have to be studied. Next slide, please. Measures of a success and uncertainties. Long-term success would be measured sort of in the typical way of, can viable bat populations be sustained? Can populations be sustained long enough to hopefully have natural resistance emerge? In the short term, we look at just, can you develop these single short interfering RNAs and pass toxicity trials, some treatment trials, and environmental analyses. And then yet the uncertainties are still many. Which genes do you really target? We've selected a few, but are those the best? What is the method of application, investigating non-target effects, off-target effects, etc.? And despite these uncertainties, I would say, despite all these caveats, I'm still invested in this because it is, as much as it may be high risk, it also is one of these few techniques that when you're facing extinction of multiple bat species throughout eastern North America that provide critical economic and ecological services for agriculture and forestry, these are solutions that do have the potential to provide continental scale protection for some of these bats in ways that most of the other solutions that we can even think of couldn't possibly be reach that kind of efficacy level, simply because it's so hard to reach most bat populations. And yet if you can get this into a small number of bat populations and they can spread it, the fungus gets spread from bat to bat. Bats know how to find other bats. It does have the potential for being able to move through populations. Next slide. I think this is my final slide. The concluding message is, just to sum some of this stuff up, that technology, these are changing daily, and keeping up with the details of every technique is just about impossible. But I would say, don't let that dissuade you from figuring out these techniques and trying to put them into these general classes, because these general classes and general principles are often the same, or at least very similar. Someone may come up with a new version of CRISPR or the next CRISPR or a new whatever, but it's still a genome editing technique. And that gives you, if you know what box to put it in, that can be very useful. Terminology, while also confusing, can be, once you learn it and learn some of the general things, that can be useful across techniques and gives you the ability to start asking these questions, trying to get at judging risk versus value, which again, I hate to even touch that subject because I know just how vast it is, and I would just simply say that it's extraordinarily complex, and that most reviews where people have gotten together in large numbers, such as the National Academy's report on gene drive on the horizon, have come to conclusions that more information is needed. And I know people always hate to hear that because some of the problems that face us are pressing. And it's very painful to hear this more information is needed. And yet that is, I think, right now for many of these things, we're at the proof of concept stage, or for some of these things, not even quite at the proof of concept stage. And we do need to know more before proceeding with some of these things, because there's no real fallback. For some of these, once released, without a kill switch, there's no real putting the genie back in the bottle. So determining risk versus value will inevitably then come down to also individual circumstances. Are you working on an island or a mainland? The techniques that you're thinking about using, and are there other related species nearby, et cetera, and all of these particular details that may influence whatever organism, whatever species that you're particularly interested in, and whatever challenge it may be facing. Those details are going to be hugely important. And I believe that's it. I believe I've left still a little bit of time for questions here. So if we advance to the next slide, I think that's the final slide, and maybe I'll turn it back over to Jonathan.
Great. Thank you very much, Dan. Thank you for that excellent presentation. Really appreciate you taking the time to share this information with us. We've already received one question for Dan in the chat box, and certainly invite those of you who have questions to type them in the chat box. We'll try to get through as many as we can in the little bit of time that we have remaining for our webinar this afternoon. The first question comes from Jeffrey Mabey. It's, is there any evidence that gene manipulation can alter natural mutation rates? And this was in regards to the slide about CRISPR-Cas9 and the possibility for unwanted mutations tagging along.
I don't know that there's any sort of a priori reason to think that they would affect. It could be set up in a way, if it affects something like DNA repair mechanisms, of which DNA repair mechanisms are incredibly complex and often highly redundant. If you set up a gene drive that's either in a place or I don't know of any examples where that's happened, that it specifically has increased mutation rates, and yet one would want to be careful and thoughtful and make sure that the organisms you selected in terms of placement, that you aren't changing, you haven't affected some of these other systems that are also in place to repair DNA. So while I put that in there, I wouldn't say it's necessarily an increased risk. I would say it's just mutations can potentially happen at a baseline rate, and so that potential could happen. If you set up things in sort of the wrong way, you could do something that might have that effect, that could affect something like DNA repair machinery and increase the rate at which you have mutations. But I wouldn't see that as something that would happen easily, and I would think that that's something that most people would generally look out for when they're coming up with their constructs or screening once they've been able to move these things, make the changes, look at how it integrated into the genome, and check for that.
Great. Thank you, Dan. Question here now from Michelle Varant. what is the likelihood of resistance evolving against gene editing technologies? Are there pressures, for example, from natural selection?
Yeah. Oh, that's such a great. Some people think it's very high. In the mosquito world, that's the perfect. That people. There have been a couple of commentaries recently, and again, I don't know if this is based on evidence or if it, but a couple of commentaries recently about mosquitoes and that the high probability of failure. They think that people are worried about these gene drives taking off like mad. And what some people envision is that, in fact, it will be a spectacular failure in terms of getting things to drive through a population is much harder than we think. Biology has gone to great lengths to keep bad things from moving through a population. And there's people who have written quite eloquently in terms of mosquitoes about why they think resistance to these gene drives will evolve quite quickly. Because, again, you're dealing with fast reproducing organisms, again, often have ability to change and edit DNA that we don't even understand. And once you've got a really deleterious thing in your genome, there may be other mechanisms out there that we know very little bit about that may deactivate or compensate for those, so that the populations that are resistant or don't pick up that gene drive might take off quite quickly. So I think that's definitely a possibility, and in some ways it's kind of interesting because it's the opposite of the concern that once you release, in some ways, given just how good biology is at sustaining itself, I would be amazed in a wild population, and I could be completely wrong, but I'll be amazed if and when they are ever able to show that they can truly drive a population to extinction in this manner, in that I think that the ability to evolve some way around it is a potential. And what it may do, though, is create population crashes. And maybe that's better. Maybe you're not dealing with extinctions, but it's something that you could introduce into the population, make the population crash, get it to such a low level. And you also know that if you can get populations to low levels, stochastically you have a higher probability of that species going extinct, perhaps, say, on an island. So it may give you that, like, say, in New Zealand with mice. Maybe if you can't even get the gene drive to really take off and just run all by itself throughout the entire island, if you could make population crashes happen over and over and over, you could get it to happen enough that at some point, statistically, some of these populations do go extinct. But that's a great question.
Great. Thank you, Dan. One more question here from Tricia Johnson. Is there any evidence of genes transferring to another organism using any of these techniques?
Well, we know with basic genetic engineering techniques that genes that you move into one organism, that if they can hybridize with other, say, weed species, if you move it into an agricultural species and they're closely related weeds in the area that they can hybridize with, those genes, there is evidence that genes have been able to move. With these specific newer technologies, I don't know that that's been shown, but it's just a similar concern. And really it all comes down to, is there a species nearby that will be able to be to some degree in the same gene pool as that species? So I don't know with these newer techniques that anyone has documented that kind of thing happening. But based on what we know from previous experience with just classical genetic engineering, it's the kind of thing that it's not just theoretical. People have been able to show that if you do have close relatives, but then it is that question of, do you have close relatives? Do you know that there are populations nearby that your plant or your mouse or your insect can interbreed with, and you don't want that happening? Now, for this, it would be unlikely, I would think, or low probability that you're going to get jumps across very distantly related species. That seems less likely, but if it's something that you could, like willow species, if you want to move it into one willow and willows are hybridizing like mad, it's the kind of thing that would be a significant problem, because it will definitely not stay just in one willow. It's going to move to any of the different groups of willows that willow can potentially hybridize with.
Great. Thank you, Dan.
Hopefully that answers.
Yep. We have time for one more question before we hit a hard stop at the end of the time for our webinar today. And a question here again from Jeffrey Maybe. Do you know of any attempts to apply these techniques to Batrachotrichium dendrobatitis, the fungus that causes chytridomycosis in amphibians?
That's a great question. I don't off the top of my head. That doesn't mean there aren't people out there working on it. And I don't. So the Kittredge disease of frogs and amphibians is incredibly a sad, terrible story, and I'm not sure if there are groups that are. I'm sure there are groups that are thinking about it. Thinking of Tim James as mycologist that in the U.S. or other places, and Matt Fisher in the U.K. Who work with Kittredge. I'd like to follow up on that and try to get back to you because I am curious. That I'm sure it's something that has been put out there of, oh, this would be we should do something like this. But I don't know if there are any funded projects to actually say. And part of the problem with Kittredge fungus is that it's incredibly, the genetics of it's hard to work with. It tends to be a diploid, which is unusual for a lot of these fungi. And we understand very little about how their genetics and genomics. And so manipulating some of those things might be difficult, but I would be curious if there are funded proposals or even grant proposals or just even labs out there that are thinking about it, because that is one Kittredge disease, as well as then now as it starts to come in the salamander, the one that affects salamanders, again being another a huge issue and one that would be would definitely, I'm sure people are thinking about it.
Yes, and Monica actually texted me here in the box saying we can ask Dee Dee Olson and also Priya Nanjapa from AFWA, who has been working a lot on both the frog chytridomycosis and the salamander fungus as well. But thank you very much, Dan. It's been a fantastic presentation. Unfortunately, we're out of time for today's webinar, but I want to thank everyone for participating. We had a really great audience today, great presentation. As I mentioned before, this presentation is being recorded, and the recording will be available on AFWA's YouTube channel. If you Google YouTube and Association of Fish and Wildlife Agencies, you'll find this recording as well as recordings of other webinars we've hosted recently. And
the conference has been unmuted.
To conclude today's webinar, I'd like to turn it back over to our wonderful partner at the Forest Service, Dr. Monica Thomasy, for some closing words. Monica.
Thank you, Jonathan, and thank you, Dan, and thanks to the participants. If anyone listening now has a particular wildlife or natural resource topic of interest you'd like us to cover, please send your request to Jonathan Maudsley. For more information on research coming from the Forest Service, you can go to the Tree Search engine. That's TREESEARCH. And the next webinar will be in August, I believe late August, and it will be an overview on institutional animal care and use committees. Look for an announcement soon. Thank you, everybody.
Great. Thank you all very much. Thank you, Dan, and thank you, Monica. Have a great afternoon. Thank you.
Thanks.